Welcome to the Saccharomyces Smackdown, this year's highly anticipated complementation test tournament. I'm your host, Tobias.
And I'm Kira Add 1 Add 2. In today's tournament, 4 unknown tester strains of yeast face off with known yeast strains. From the results of this tournament, we'll know their true colors.
Tonight, we're breaking down epic matchups between haploid yeast strains as they cross, mate, and reveal their genetic secrets. The question on everyone's mind: would mutations complement, or will we see a red zone turnover?
Here comes our favorite players. On one side, we've got 2 haploid alpha mating type strains, HB1 and HB2. On the other side, we've got 2 haploid A mating type strains, HA1 and HA2. When mat A mates with mat alpha, they create diploid cells.
The crowd doesn't seem very interested in them.
Well, Tobias, that's because these players aren't why the crowd has come. They're here to test off the unknown players and see if they complement or not. These yeast strains have competed in the complementation test tournament so many times that the crowd knows exactly what to expect from them. After all, HB1 and HA1 carry an AD1 mutation, while the HB2 and HA2 carry an AD2 mutation.
I see, Kira Add 1 Add 2, and here comes the unknown players. XA1, XA2, XA3, and XA4 have taken the field. Now, here's how the game works. When these strains mate, we'll be looking at the phenotypes of their diploid progeny. If their colonies turn red, it's a failed complement. If they're white, it's a complementation.
That's right, Tobias. And to clarify, a failure to complement means that both the mutations are on the same gene, while complementation means that the mutations are on different genes.
Here we go. HA1 and HB1 are lining up, and we're off. HB1 crosses with HA1. Wow, that's fast. The colonies are red. That's a textbook fail to complement. Wow.
Don't get so excited, Tobias. That's exactly what we expected. Both of the HB1 and HA1 carry an AD1 mutation, so after mating, the colonies carry 2 AD1 mutations. The AD1 gene plays a critical role in converting AIR into adenine, and without it, AIR accumulates, generating red mutant colonies.
Look, HB1 is squaring off against HA2, and white. That's a clean complement. These mutations are in different genes. HB1's AD1 and HA2's AD2 are balancing each other out for the win.
Keep an eye on HB2 over there. Looks like it's getting ready to make some advances. Aha! 2 at once! Upon contact with HA1 and HA2, we've got white and red, which is the exact opposite of the HB1's crosses.
And we could have predicted those, right? Kira Add 1 Add 2? We know how mutations in the Add2 gene tend to square up against mutants in the same gene versus in a different gene. Alright, I want to see some action from the unknowns now. I've been waiting all night for this. Wait, what's that sound? Listen, the crowd is going wild. The results are in from the unknowns, but do the spectators actually know what these results mean?
Probably not. You got this one, Tobias.
Well, as for XA1 and XA2, we've got resulting red colonies when crossed with both HB1 and HB2. Failure to complement in both AD1 and AD2. No, it couldn't be. Could this mean
that both XA1 and XA2 have mutations in both the AD1 and AD2 genes? Sure, it could. And as for XA3 and XA4, their crosses look familiar, don't they? They have the same results as HA2 and XA1's crosses, respectively, so they must have the same genotypes.
Wait, but are we just going to skip over the fact that XA4 was white to start with?
Okay, fine. Let's unpack that. One thing we forgot to mention was that if yeast have a mutation in the adenine biosynthesis pathway upstream of the AD1 and AD2 genes, the cell doesn't even have a chance to accumulate AIR in the first place, and thus no red pigment.
So, XA4 has mutations in the AD1 gene, AD2 gene, and in upstream genes?
Exactly.
Alright, that's enough complementation for me. We'll see you next time.